ವಿದ್ಯಾರ್ಥಿ ಸ್ನೇಹಿತರೆ, ನಾನು ಡಾಕ್ಟರ್ ಕೆ ಎಸ್ ಕುಮಾರ್ ಅಂತೇಳಿ ಡಾಕ್ಟರ್ ಕೆ ಎಸ್ಗೆ ನಾವು ಯೂಟ್ಯೂಬ್ ಚಾನಲ್ನಲ್ಲಿ ನನ್ನ ವೀಡಿಯೋಸ್ನ ನೀವು ನೋಡ್ಬೋದು. ಈಗಾಗಲೇ ಕೆಲವು ವೀಡಿಯೋಸ್ನ ಅಪ್ಲೋಡ್ ಮಾಡಿದ್ದೀನಿ. ಇವತ್ತು ನಿಮಗೆ ಎನ್ಸೈಮ್ಸ್ ಬಗ್ಗೆ ಮಾತಾಡಬೇಕು ಅಂತ ಇಷ್ಟಪಡ್ತೀನಿ. ಡಿಸ್ಕಷನ್ ಆದಮೇಲೆ ಕೆಲವು ಇಂಪಾರ್ಟೆಂಟ್ ಕ್ವಶನ್ಗಳು ಕೂಡ ನಿಮಗೆ ಹೇಳ್ತೀನಿ. ಥ್ಯಾಂಕ್ ಯು. ಓಕೆ, let us begin with the discussion. Enzymes. What are enzymes? Enzymes are substances produced by a living organism which acts as a catalyst to bring about a specific biochemical reaction. You are all familiar with the catalyst. Catalyst is a material or substance which alters the velocity of the chemical reaction. These enzymes are the biological catalysts. That means they are able to work in the biological system. And these are the wonderful molecules which are proteinaceous in nature. They bring about chemical reaction in sometimes fraction of the seconds, which would require months together outside the laboratory. There is a lot of things in discussion. That is, let us go one by one. What is the chemical nature of the enzyme? To tell you very precisely, all enzymes are the proteins, but all proteins are not enzymes. Most of the enzymes are proteinaceous in nature. Recently, some of the non-protein enzymes are also recognized. We talk about only the proteinaceous enzyme. You can observe an equation in this here. It is the combination of apoenzyme, cofactor, which leads to the holoenzyme. That is, holoenzyme is a combination of these two things, apoenzyme and the cofactor. Here, the apoenzyme is a proteinaceous part, cofactor is a non-proteinaceous part. Holoenzyme is a biologically active form. Neither this cofactor nor this apoenzyme is able to bring about the catalytic work or enzymatic work. Only combination of these two, which is called as the holoenzyme, is biologically active. Let us see what is this cofactor. Cofactor may be a coenzyme, cofactor may be a prosthetic group, cofactor may be a metal ion. How do you distinguish this coenzyme and prosthetic group? A non-protein organic substance which is dialysable, thermostable and loosely attached to the protein part is called coenzyme. A non-protein organic substance which is dialysable, thermostable and loosely attached to the protein part is called as the coenzyme. Secondly, the prosthetic group. The prosthetic group, an organic substance which is dialysable and thermostable, which is firmly attached to the protein or apoenzyme portion of the enzyme. And thirdly, a metal ion. Metal ion may be an activator. An activator is a substance which increases the activity of the enzyme. So, activator is usually a metal ion. It increases the catalytic work, or you can say these activators are the catalyst of the catalyst. So, they increase the presence of this ion increases the activity of the catalyst or enzyme. Next, let us go for the classification of these enzymes. How these enzymes are classified, how these enzymes are named. Previously, enzymes were named based on the substrate on which they are going to act upon. When very few enzymes were known, suppose if it acts on urea, it is called as urease. If it acts on sucrose, it is called as the sucrase. Like that they used to name. But after knowing the many number of the enzymes, there is a systematic way of naming the compound with easy numbers and the classification of the enzyme. I am going to discuss only about the classification of the enzymes. How these enzymes are classified. So, these enzymes are classified into 6 classes depending upon the nature of the chemical reaction they bring about, they act upon. Okay. So, they are the oxidoreductases are the one class of compounds. These oxidoreductases, so this category of compounds bring about the oxidation reduction reaction. That is, so they transfer the hydrogen and oxygen atoms or electrons from one substrate to the another substance. So the enzymes which brings about the oxidation reduction reaction is called oxidoreductases. One of the familiar example for that category is the of course dehydrogenase oxidases. Secondly, the transferases. The transferases are the specific group from one substrate to the another. They transfer a specific group from one substrate to the another substrate. So transaminase, kinases, etc. are the examples for these transferases. Thirdly, the hydrolyses. See, the word itself says that these are the enzymes which bring about the hydrolysis of a substrate. Usually, the esterases, digestive enzymes are the examples for these hydrolyses. And fourth one is the isomerases. Here also the word says that these enzymes change of the molecular form of the substance. That means they bring about the isomerization of the substance. That is, so you can observe the examples are the phosphoxo isomerase, fumarase are the familiar examples. They bring about the isomerase. Fumarase enzyme convert fumarase to malic acid, like that. Then the fifth category of enzymes are the lyases. These are the non-hydrolytic removal of the group. These enzymes bring about the non-hydrolytic removal of the group or addition of the group to the substrate. And otherwise you can say lyases are the enzymes which bring about the elimination reaction or the addition reaction. One such familiar example in the biological system is the decarboxylases. And finally, the ligases. Ligases are also known as the synthetases, and they are involved in joining of two molecules by the formation of the new bonds. So they are also called as the synthetases because they are going to synthesize this one. They are going to link the two or more substances together. One such example is the citric acid. Citric acid synthetase is an example. Totally, the enzymes are classified into 6 classes. So oxidoreductases, transferases, hydrolyses, isomerases, lyases, and ligases. So this is a short table which is going to give information regarding the class of enzymes and the enzyme, the reaction which is catalyzed by those enzymes, and also example for each class. Let me see some more example to these classes. You are going to see the enzyme that is lactate dehydrogenase. This is one of the enzymes which bring about the oxidation reduction reaction, that is a transfer of the hydrogen. The pyruvate becomes the lactate. So the pyruvate is actually reduced to lactate. So this bring about the oxidation reduction reaction. So lactate dehydrogenase is an example for the oxidoreductase. The next is the transferase. This is aspartate amino transferase. You can remember this. So this amino group from the aspartate is for transfer to the alpha keto glutarate. It becomes a glutamate, and this enzyme is involves in the transfer of amino group from one substrate to the other substrate. You can observe one more example here, that is a hydrolase. That is going to bring about the hydrolysis. You can see here that is the hydrolysis of the material. Even the urease comes under this category. Urea is hydrolyzed to give the ammonia and carbon dioxide with an enzyme called urease. Here it is acetyl, that is acetyl that is one of the enzyme which is involved in the hydrolysis of the reaction. And the fourth one is the lyase. Here you can see the addition of the water to the cis-accotinase and to give the isocitrate. And this is the reaction observed in the Krebs cycle. Another class of enzymes are the isomerases. Here you can say isomerases bring about the isomerization of the reaction. That is the phosphate, that is glucose 6-phosphate becomes the fructose 6-phosphate. Alpha glucose 6-phosphate is converted into fructose 6-phosphate. This is a reaction, important reaction that is observed in the glycolysis. Then lastly, you can see that ligase reaction, that is the synthetic reaction. that is tyrosine tRNA synthetase is an example for this ligase. Here it is going to link or the attached or the join the ATP and tyrosine molecule with the help of the transfer RNA, and L-tyrosine transfer DNA, that is plus of AMP plus inorganic phosphate is formed. These are the different examples of the different classes of the enzymes. Next, let us see what is an active site. An active site is the region of an enzyme where the substrate molecule bind and undergo a chemical reaction. The active site is a portion on the surface of the enzyme where exactly there will be a binding of substrate molecule on its surface. It's a small portion on the surface of the enzyme which is actually involved in the catalytic work. So what exactly that active site? The active site consists of amino acid residues that form temporary bonds with the substrate and residues that catalyze the chemical reaction of that substrate. And of course, so the active site occupies only 10 to 20% of the volume of the enzyme. And of course, it is most important part of the enzyme because it is the part which is going to catalyze the chemical reaction. Usually this active site consists of 3 to 4 amino acids, while other amino acids within the protein are required to maintain the tertiary structure of the enzyme. So usually the active site consists of a bunch of side chains of different amino acids. Of course, out of those bunch of side chains of amino acids, and some of them are involved in the binding and some of them are involved in the catalytic work. You can see here in the other side. So this is the size of the protein molecule, and here it is, which is in the color, this portion is the active site. So compared to the size of the active site, size of the protein is more. A small portion on the surface of this enzyme actually does the catalytic work. This is a three-dimensional view of the active site of the enzyme. Then we shall understand what is meant by the specificity of enzyme. Specificity is the ability of an enzyme to choose exact substrate from a group of similar chemical molecules. The specificity is actually a molecular recognition mechanism, and it operates through the structural and conformational complementarity between the enzyme and substrate. To tell you very briefly, enzymes, since they are proteinaceous in nature, they are very, very specific in their action. So they choose the substrate and bring about the chemical changes. Then let us see what are the different types of specificity. Specificity, importantly, we have other class of specificity also. Importantly, we can classify the specificity into 3 categories. That is, one is absolute specificity, and the other one is the group specificity. Third one is the stereochemical specificity. Absolute specificity, you can the enzyme will catalyze only one reaction. This enzyme under any circumstance do not act upon any other substrates. And group specificity is one. The enzyme, these sort of enzymes will act only on molecules that have specific functional group, such as amino group, such as phosphate group, such as methyl group. And these class of enzymes, that is group specificity enzymes, are going to act upon the specific functional groups. They won't choose the entire substrate. If they contain that substrate group, they are going to act upon. And thirdly, the stereochemical specificity. Stereochemical means maybe cis form, trans form, L form, D form, like that. So these sort of enzymes will act on a particular stereo or optical isomers. They are going to act upon a particular stereo or optical isomers like this. So these optical isomers means L isomer, D isomer, like this. So importantly, we have 3 categories. Absolute specificity is exhibited by an enzyme which act only on the one particular substrate. Group specificity is exhibited by the enzyme which is going to act upon a particular functional group. And stereochemical specificity is observed by an enzyme if it acts on a particular stereoisomer. So that is the different types of specificity. Let us see some of the different examples for this different specificity. So that is an enzyme called, that is fumarase, is going to exhibit the absolute specificity. It acts on the trans isomer, it never acts on the cis isomer. This enzyme is going to act upon only the trans isomer, not upon the cis isomer. Here you can say. So it is going to act on only the trans isomer, never acts on the cis isomer. Here you can see here there is another enzyme called pepsin. It is going to catalyze all bonds which are all amino acid linkage, that is the peptide linkage. Pepsin is going to break the peptide bond. It is irrespective of the nature of the amino acid which is attached. So the bond only it looks, and it breaks only the peptide bond. And thirdly, stereospecificity you can observe. So one of the example for that stereospecificity is this one. So here, so L isomer, L alanine oxidase enzyme, that is going to act only on the L alanine. It never acts on the, of course, that is the D alanine. So it acts only on the L alanine, never acts on the D alanine. So it is going to choose the particular stereoisomer. Next, let us see something regarding the mechanism of the enzyme action. So how exactly the enzyme and substrates are going to interact with each other? We have two important theories to explain the mechanism of the enzyme action. They are lock and key model theory proposed by the M.L. Fisher and induced fit theory proposed by the Coastland. I will tell you the few points regarding the lock and key model theory. According to the lock and key model theory of Fisher, the enzyme is a rigid molecule. That means it possesses a rigid shape, and it has got the structural complementarity with the substrate. Of course, and that means the enzyme and the substrate have got the complementary structure with each other. And during the enzymatic process, during the chemical reaction, these two structures you can observe. This is the substrate structure, this is the enzyme structure. Here these two shapes are complementary to each other. Because of this complementarity, they combine and forms the enzyme substrate complex. This enzyme substrate complex decomposes to give the product and enzyme back. This theory highlights that this enzyme is a rigid molecule. The substrate and enzyme have got the structural complementarity. And this theory explains why certain enzymes show the absolute specificity. So other mechanism is the induced fit theory proposed by the Coastland. This theory is quite opposite to the lock and key model theory. According to this theory, enzyme is not a rigid molecule, but it is a flexible molecule. It changes its shape during the chemical action. It changes its shape. You can see here the shape of the substrate. Shape of the substrate is not complementary with the shape of the enzyme. So enzyme is a flexible molecule. This flexible molecule is going to adjust itself. It's going to change its shape and fit into the enzyme. It forms the enzyme substrate complex. This is a quite opposite theory. So here enzyme is not a flexible molecule, not a rigid molecule, but it is a flexible molecule. Whenever it comes in contact with substrate, it changes its shape and forms the enzyme substrate complex, which is going to decompose to give the product and enzyme back. These are the two popular theories to explain the mechanism of the enzyme action. Those theories are called lock and key model theory and induced fit theory. Next, and of course, the very important thing, what are the factors that influence the velocity of the chemical enzyme catalyzed reaction? I highlight only the four factors that influence the velocity of the enzyme catalyzed reaction. The first factor is the substrate concentration or concentration of the substrate. You can observe in this first graph that the velocity of the enzyme catalyzed reaction increases in the beginning and finally becomes constant. That means, so as the substrate concentration, the velocity of the enzyme catalyzed reaction increases and it is proportional. After some time, it remains constant or it reaches the maximum value. So this is because enzyme gets saturated, and the maximum velocity is observed. Here you can see this one. So why there is a saturation of the enzyme and all those things. Usually there will be interaction between the substrate and the enzyme, and the substrate concentration, if you increase, the chances of formation of the enzyme substrate complex increases, and if you keep on increasing, the velocity increases. When it reaches the maximum, even if you increase the substrate concentration to the greater extent, the enzymatic reaction rate or velocity remains constant. Okay, this is another plot which shows the variation of the velocity and the substrate concentration. Here you can see some of the things in this second graph, that is the maximum velocity attained by the enzyme catalyzed reaction is called V maximum. And if you take the 50% of the V maximum and if you draw a line to this curve and that is, if that line is extended to the x-axis, that is the substrate concentration, that represents another constant that is equal to Km, that is the Michaelis Menten constant. So you can closely observe this graph. The graph increases, the maximum velocity is called V maximum, 50% of the maximum velocity is called half V maximum. If you extend this line to the curve and corresponding substrate concentration is the Michaelis Menten constant. Let us understand what is this Michaelis Menten constant, how it can be obtained, and what is the significance of this Michaelis Menten constant. Here, that Michaelis Menten constant can also be obtained by the Lane-Vievers-Burke plot method or Lane-Vievers-Burke equation or double reciprocal graph. You can observe in this graph, so in y-axis, the reciprocal of the velocity is plotted. In x-axis, the reciprocal of the substrate concentration is plotted. The graph will be like this. And of course, it touches the y-axis, which is equal to 1 over V maximum. So that point gives you 1 over V maximum, the reciprocal of the V maximum. And if you take the slope of this line, it will give you the ratio of the Km to V maximum. And if we extend this straight line to the x-axis, it meets the x-axis which is equal to minus 1 over Km. These are the important things that is related to the Lane-Vievers-Burke plot method. This is one of the methods from which Michaelis Menten constant can be determined. Let us see what is that Michaelis Menten constant and how it is related to the other things. This, of course, it is a useful graphical method to analyze about the enzyme catalyzed reaction. That is, that Michaelis Menten constant can be obtained by this equation. You can observe here the Michaelis Menten constant equation. So here you can see that one, V is equal to V maximum multiplied by the substrate concentration divided by Km, that is Michaelis Menten constant, plus the substrate concentration. We have the derivation of this equation also. Let us not much bother about the derivation, where V is the initial velocity, V maximum is the maximum velocity, and Km is the Michaelis Menten constant, and S is the substrate concentration. I importantly stress upon the significance of this Michaelis Menten equation. And of course, this Michaelis Menten equation relates substrate concentration with velocity of the chemical reaction, and it gives quantitative calculation of enzyme characterization, and also it is going helpful to analyze the enzyme inhibition. Of totally you can say this Michaelis Menten constant is going to give information regarding the binding of the enzyme and the substrate. Next, we shall go to another factor, that is the concentration of the enzyme. This graph will explain you very clearly the rate of the enzyme catalyzed reaction is going to increase like this if you increase the concentration of the enzyme, or you can say the rate of the enzyme catalyzed reaction is directly proportional to the concentration of the enzyme. And of course this is very helpful for the determination of the enzyme concentration in the clinical application, and of course for diagnosis of inborn errors in the metabolism. So that is another significance. Next one, these two factors are going to affect the rate of the enzyme catalyzed reaction to the significant extent. So temperature. See, this graph is very useful to explain you the effect of temperature on the rate of the enzyme catalyzed reaction. In my axis I am plotting the enzyme activity, and the x-axis temperature is plotted. In the beginning, that is at the lower temperature, the rate of the enzyme catalyzed reaction is less. If you keep on increasing the temperature, the rate of the enzyme catalyzed reaction increases, and after reaching certain temperature, it falls down. That means there is an optimum temperature. So the temperature at which the enzymatic activity is maximum is called optimum temperature. Why this observation is observed? At low temperature the enzyme is inactive. At high temperature again the enzyme is inactive. At certain temperature, which is known as the optimum temperature, the enzymatic activity is maximum. Usually the optimum temperature for most body enzymes is at the 37 degree Celsius. So at low temperature as well as the high temperature do not favor the ability of the enzyme to catalyze the chemical reaction. Okay. So the reason is also very much clear. So enzymes are proteinaceous in nature, and of course at low temperature they undergo denature, and at high temperature also they undergo denature, and because of this reason also there is a variation like this. And finally, so the pH value, that is the pH, you are very familiar, they are pH sensitive, enzymatic activity is pH sensitive. In the graph you can observe here, so similar to the temperature curve, so at lower pH value the enzymatic activity is less, at higher pH value also the enzymatic activity is less, and at certain pH value which is near to the neutral the enzymatic activity is maximum. The pH at which the enzymatic activity is maximum is called optimum pH. Usually this optimum pH is around 7, and of course each enzyme maintains its optimum pH value, so they are active in that particular pH range. And again the reason is also same. At lower pH the enzyme is inactive because it is proteinaceous in nature. Even at very low pH and very high pH bring about the denaturation of the enzyme, and therefore they are inactive in nature. The next, the final thing, that is about the inhibitor. What is an inhibitor? What is the inhibition? The prevention of an enzyme process as a result of interaction of inhibitor with an enzyme is called inhibition. So inhibitor is in any substance that can diminish the velocity of an enzyme catalyzed reaction. Okay, so this inhibitor is a material which reduces the activity of the catalyst. So I will tell you, I am showing the a slide here. Here, this material is an inhibitor, whereas this substrate. And this inhibitor molecule stops the substrate to join the active site, and therefore the enzymatic activity is reduced. So in short, I repeat it once again. Any substance that can diminish the velocity of an enzyme catalyzed reaction is called an inhibitor. The process phenomenon is called as the inhibition. Next, we shall see what are the types of inhibition. We have two types of inhibition, that is a competitive inhibition and non-competitive inhibition. So, to tell you in a very brief way, so in this competitive inhibition, the inhibitor compete with the substrate for the active site. So, inhibitor competes with the substrate for the active site, and formation of the enzyme substrate complex is reduced, while instead of that, formation of the enzyme inhibitor complex is formed. So, in competitive inhibition, you can observe here, so the inhibitor molecule competes for this active site with the substrate, and thereby it reduces the formation of the enzyme substrate complex. Next, we shall see. So, that is an example for the competitive inhibition, that is statin drug as example of competitive inhibitor. So, the statin drugs such as Lipitor compete with HMG-CoA, so, and inhibit the active site of HMG-CoA reductase. Another kind of inhibition is a non-competitive inhibition or uncompetitive inhibition. In this type of inhibition, inhibitor does not compete with the substrate for the active site of enzyme. Instead, it binds to another site known as the allosteric site. Here, exactly the inhibitor molecule do not compete for the active site. So, inhibitor molecule sits somewhere here, but somehow it sees that the substrate molecule do not come in contact with the enzyme molecule. There is no competition for this active site, but somehow it prevents the formation of the enzyme substrate complex. That inhibition is called as the non-competitive inhibition. And of course, these are the examples for the non-competitive inhibition. The drugs to treat cases of poisoning by methanol or ethylene glycol that act as a non-competitive inhibitor. Of course, tetramethylene sulfoxide and 3-butyl thiolene 1-oxide are the non-competitive inhibitor of liver alcohol dehydrogenase. So, the liver alcohol dehydrogenase enzyme is inhibited by the presence of this material, that is tetramethylene sulfoxide and 3-butyl thiolene 1-oxide, etc. So, that's about the inhibition of the enzyme. Thank you. And I am going to share some of the important questions that are possible in this chapter. Thank you. ಸ್ನೇಹಿತರೆ, ಇಷ್ಟೊತ್ತು ನಾನು ಎನ್ಸೈಮ್ಸ್ ಬಗ್ಗೆ ನಿಮಗೆ ಕೆಲವು ಮಾಹಿತಿನ ಹೇಳಿದ್ದೀನಿ. ಇದು ಪರೀಕ್ಷೆಗಳಲ್ಲಿ ಎಂಥ ಪ್ರಶ್ನೆಗಳನ್ನ ಕೇಳ್ಬೋದು ಅನ್ನೋ ಒಂದು ಅನುಮಾನ ನಿಮಗೆ ಇರುತ್ತೆ. ಯೂಶಲಿ ಈ ಚಾಪ್ಟರ್ನಲ್ಲಿ ಏನು ಪ್ರಶ್ನೆ ಕೇಳ್ತಾರೆ ಅನ್ನೋದನ್ನು ಒಂದೆರಡು ಬಗ್ಗೆ ಅದ್ರ ಬಗ್ಗೆ ಒಂದೆರಡು ಮಾತಾಡ್ತೀನಿ. ಇದರಲ್ಲಿ ತುಂಬ ಬೇಸಿಕ್ ಆದ ಪ್ರಶ್ನೆಗಳು: ವಾಟ್ ಆರ್ ಎನ್ಸೈಮ್ಸ್, ಹೌ ಆರ್ ದೇ ಕ್ಲಾಸಿಫೈಡ್. ಸೊ ಯು ಹ್ಯಾವ್ ಟು ಗಿವ್ ದಿ ಡೆಫಿನೇಷನ್ ಫಾರ್ ದಿ ಎನ್ಸೈಮ್ and you have to give the detailed classification of all the enzymes, that is 6 classes: oxidoreductases, transferases, hydrolases, lyases, ligases, like this. You have to give all the classification. And what are enzymes? Give the classification of enzymes and give example for the different all the classes of the enzymes. So that may be a big question for you. Then after that, what is the chemical nature of the chemical composition or chemical nature of the enzymes? There you have to include the cofactor, coenzyme, everything. Then you may be asked to give the definition of the active site. What is an active site? Give the structural features of the active site. And you may be asked to go for the different types of specificity. What is specificity of enzymes? Give the different types of specificity enzymes and give example to each class. Then after that, you may be asked to go for the mechanism, illustrate the mechanism of enzyme action. There you are supposed to explain what is Lopen-Key model theory of Emil Fischer and what is induced fit theory of Koshland. You are supposed to explain. And next thing is regarding the factors influencing the velocity of the enzyme catalyzed reaction. They may ask you to write mention the factors that influence the velocity of the enzyme catalyzed reaction. They may ask you to explain how the substrate concentration, how the enzyme concentration, how the temperature, pH influences the velocity of the enzyme catalyzed reaction. And finally, regarding the an enzyme inhibition, that is the what is an inhibitor, what are the different types of inhibition, what is a competitive inhibition, what is a non-competitive inhibition, and give example to each category. Okay. Thank you. These are the possible questions. In the substrate concentration influence on the enzyme catalyzed reaction, you may be asked to go for the Michaelis Menten equation. You may be asked to write the Michaelis Menten equation and significance of the Michaelis Menten equation also. Thank you very much. If you like this video, give like to this video and subscribe to my channel.